Okay, let's unpack this. When we first encounter genetics, the foundation is usually Gregor Mendel, right? And his perfectly organized peas.
Yeah, dominant, recessive, nice, predictable ratios. Seems simple enough.
But the second you step out of that, the monastery garden and into the real world, inheritance gets infinitely more complex. It really does. Today, we are undertaking a deep dive into that true complexity, moving far beyond just those two basic principles Mendel established. Our mission here is to synthesize the sources that explain why biological variation is just so abundant. We're focusing on the surprising roles of interacting genes, non-nuclear DNA, and the powerful environmental signals that shape development.
Exactly. That's the core theme running through the material we looked at. Our sources reveal that inheritance isn't just governed by simple dominant recessive nuclear genes. It's really governed by a complex matrix. Think intricate gene interactions, what we call epistasis, combined with factors outside the nucleus, often from the mother, and these profound environmental cues. And all of this complexity creates this staggering amount of genetic and phenotypic variability. That's ultimately the raw fuel for evolution.
So if you want to understand the true mechanisms that generate the sheer diversity we see in life, especially how genes and the environment kind of cooperate and sometimes conflict to shape fundamental features, including something as crucial as sex, well, stick with us. Okay, so you have to start by acknowledging that dominance itself is rarely just all or nothing. What happens when the heterozygote, the AA individual, expresses a trait that's genuinely intermediate between the two parents? Not quite one, not quite the other.
You're describing incomplete dominance. A classic example, and easy to picture, is flower color. You cross a plant with, say, red flowers, that's AA, with a plant with white flowers, AA. The offspring, the AA ones, won't be red or white, they'll actually be pink.
Kink, a shrew intermediate blend.
Exactly. It's an intermediate blending of the characteristics.
And it's not just in these obvious visual cases like pink flowers, is it? I mean, even traits Mendel thought were discontinuous, clear-cut, like smooth versus wrinkled peas, kind of break down when you look closer.
They absolutely do. When we assess those same P traits, but at a much higher resolution, looking at things like the number of starch granules or maybe enzyme concentrations, the heterozygotes show intermediate values. So what looks like simple dominance at the level of seed texture is actually incomplete dominance at the biochemical level. Depends on how closely you look. And this idea, this phenomenon of different dominance levels within one trait is really starkly visible in humans with sickle cell anemia.
Okay, how so?
Well, at the level of whether or not you have the full-blown disease, the trait is recessive, right? You need 2 copies of the allele.
Right.
But if you look at the level of the red blood cell shape itself, it's considered incompletely dominant. Heterozygotes have some abnormal sickle-shaped cells, but they aren't fully symptomatic like individuals with 2 copies.
So intermediate again.
Intermediate again. And here's where it gets really interesting. At the molecular level, looking specifically at the production of the hemoglobin protein, the 2 alleles are actually
Codominant. Is that where both are fully expressed at the same time? Can you define codominance and multiple alleles for us, especially thinking about how they add to variation?
exactly codominant. So first, a multi-allelic trait just means a gene has more than 2 possible versions or alleles in the population. The sources point out human alpha and beta globins. For instance, they have hundreds of known alleles.
Wow, hundreds.
Yeah. And codominance happens when a heterozygote expresses both functional alleles simultaneously, not a blend. So the sickle cell heterozygote makes both the mutant beta globin protein and the normal beta globin protein. We see the same thing with the ABO blood group antigens. If you have the A allele and the B allele, you don't get an intermediate blood type. You express both A and B antigens. That's codominance.
Okay, so that's alleles interacting within one gene. Now let's move up a level. How do different genes interact? This is where epistasis comes in, right?
Epistasis, yes. A really foundational concept for understanding complexity. Basically, epistasis is when one gene fundamentally changes the qualitative phenotypic effect produced by a completely different gene.
It modifies the outcome of another gene.
Precisely. It allows for complex, multi-step processes like how pigment gets produced.
Yeah.
One gene might make a precursor, another modifies it, another transports it. If any step is blocked by a particular allele, it changes the final color, regardless of the other genes. But what's truly eye-opening, and the sources really emphasize this, is the sheer potential for variation this interaction creates.
Yeah, the numbers mentioned are just mind-boggling.
They really are. Think about it. If a single gene has just 4 alleles, That already gives you 10 different possible diploid combinations in individuals.
Okay.
Now extrapolate that. If you have just 100 genes like that in an organism, which is a tiny fraction of the total genome, you can get 10 combinations. That's 10 billion possible genotypes.
10 billion from only 100 genes interacting.
An absolutely astronomical number. And that staggering potential genetic variability, driven largely by these gene interactions, is the raw material, the starting point for natural selection to act upon. It's also interesting, the sources note that viruses with their much simpler genomes tend to show fewer epistatic interactions compared to complex eukaryotes like us. This suggests that this intricate web of interactions is itself an evolved feature, perhaps allowing for more complex adaptations.
Okay, so we've established how this internal gene wiring, epistasis, and dominance variations creates this huge potential for variation. But that variability doesn't all originate neatly in the nuclear DNA, does it? How do we account for extranuclear inheritance?
Right, that's another major deviation from the simple Mendelian nuclear rules. It's often called cytoplasmic inheritance. And it arises because certain organelles within the cell, specifically mitochondria and chloroplasts in plants, actually have their own genetic material, their own small circle of DNA, completely independent of the main chromosomes in the nucleus.
And why does that matter so much for inheritance patterns?
It matters profoundly, especially in animal development. The sources mention verbally referencing figure 13.2 here, which illustrates this. Basically, when fertilization occurs, almost all the cellular components for the new zygote, we're talking the cytoplasm, the mitochondria, the ribosomes, even the initial microstructures that guide early development, they're all contributed almost exclusively by the maternal egg.
The egg provides the whole workshop, essentially.
Pretty much. The sperm delivers the paternal nuclear DNA, but very little else. So any traits encoded in the mitochondrial DNA, mtDNA, are inherited maternally. And because this mtDNA isn't subject to the shuffling of recombination like nuclear DNA is, it tends to show significantly less variation over time. This makes it an incredibly critical tool for scientists trying to reconstruct ancient evolutionary history and relatedness. Less shuffling means a clearer historical signal.
That classic maternal inheritance story is fascinating, but it can still yield some real surprises, can't it? I was genuinely shocked to read about the Ambystoma maculatum, the spotted salamander.
Oh yeah, that's a wild one. Researchers discovered green algae actually living inside the cells of the salamander embryos, the larvae, and even the adults, acting as an endosymbiont.
Algae living inside of vertebrate cells.
Exactly. It totally opened up a new perspective on previously unknown kinds of vertebrate endosymbiosis. It shows just how tightly integrated maternal inheritance pathways can sometimes be with the immediate environment, even incorporating other organisms.
Okay, wild indeed. Now let's swing back to the nucleus and focus on the main engine of variation itself, sexual reproduction. The sources highlight two key factors that come from sex: recombination and something called gene linkage.
Right, so gene linkage just refers to the fact that genes located close together on the same chromosome tend to be inherited together, as a block. They're physically linked. However, recombination, which happens through the physical process of crossing over during meiosis, where homologous chromosomes exchange segments, actively shuffles these linked genes, it breaks up those blocks. In humans, this recombination process alone can produce literally millions of different genetic combinations in the eggs and sperm.
Millions of combinations from one individual.
Yes. And this variation, generated by shuffling existing alleles into new combinations, is thought to be absolutely key to long-term survival, especially in changing environments. It's generally why species that reproduce purely asexually, just cloning themselves, usually have higher extinction rates over the long haul compared to sexually reproducing species. They lack that rapid generation of new combinations.
Okay, so if sex is so vital for generating variation, but it also requires, finding a mate, courtship, all that energy investment, why exactly does it persist so strongly? What's the primary benefit the sources seem to land on?
Yeah, that's one of the great ongoing questions or paradoxes in evolutionary biology. Why sex? There are several hypotheses discussed, like maybe it provides better DNA repair mechanisms, or maybe it helps populations adapt faster in highly variable environments.
It makes sense.
But the best supported reason, according to the sources, for the widespread persistence of sex seems to be its effectiveness in removing deleterious or harmful mutations from the population.
Ah, the cleanup mechanism.
Exactly. Studies, like those mentioned on the water flea Daphnia pulex, which can reproduce both sexually and asexually, show that harmful mutations tend to accumulate much faster during asexual reproduction cycles. Sex, through recombination, can separate good genes from bad genes, allowing selection to more effectively purge the bad ones. It also helps beneficial mutations spread more quickly by combining them together in one individual. So talking about sexual reproduction leads us straight into how sex itself is determined. And broadly speaking, the mechanisms fall into two main camps: sex chromosome-based determination, like in us, and environmentally induced sex determination.
Let's tackle the genetic masters first, like the familiar mammalian XXXY system. What is the absolute minimal genetic switch that, kicks off male development in mammals?
That would be the SMAG gene. It stands for the sex-determining region Y gene. It sits on the Y chromosome, and it produces a protein called the testis-determining factor. Think of it as an upstream signaling gene. It doesn't build the testes itself, but it initiates the whole developmental cascade that leads to male characteristics.
The master switch.
A master switch. And its fundamental role was proven pretty dramatically back in 1991. Researchers took genetically female mice, XX, and just transgenically introduced the spray gene into them.
And what happened?
They developed as males. Physically male, despite having two X chromosomes, that single gene was enough to flip the switch.
Wow. Okay, so despite the power of that tiny spray switch, the Y chromosome itself seems, well, surprisingly fragile over evolutionary time, doesn't it?
It is remarkably fragile, evolutionarily speaking. If you compare the human X and Y chromosomes, the X is huge, about 165 megabases of DNA, carrying around 1,000 genes. The Y chromosome is much smaller, maybe 60 megabases, and contains only about 50 functional genes. It's clearly degenerated over time.
Why? Why does it shrink and lose genes?
A major reason is its restricted ability to recombine. Most of the Y chromosome doesn't have a homologous partner to pair up and swap segments with during meiosis, unlike the X, which pairs with the other X in females, or autosomes, which pair with their homologs. This lack of recombination limits the effectiveness of DNA proofreading and repair processes. So mutations, especially harmful ones, tend to accumulate much faster on the Y chromosome than on the X or autosomes, leading to gene loss and decay over millions of years.
And this degeneration process can lead to some pretty extreme examples outside the human lineage, right? Where the Y is just gone.
Absolutely. The sources point out some fascinating cases. There are two species of mole voles, genus Alobius, and two species of Japanese spinous rats, genus Tokudaya, where the males have completely lost the Y chromosome altogether.
No Y chromosome at all. So they're exomales. How do they determine sex?
Exactly, exomales. The necessary sex-determining genes, likely including something equivalent to SROI or its downstream targets, have actually translocated, moved onto other chromosomes, the autosomes. It shows how quickly and radically the fundamental genetic basis of sex determination can evolve.
It really does. And outside of mammals, the diversity just explodes. We see XX female exomale systems in nematodes like C. elegans. Then there's the Z's male ZW female system common in birds and snakes.
Right, where the female is the heterozygotic sex, ZW, instead of the male.
And then there's the platypus, which the sources mention has this bizarre system with 5 X chromosomes and 5 Y chromosomes.
Yeah, the platypus system is wild. 10 sex chromosomes. And intriguingly, its X chromosomes show links to ancient reptilian sex determination systems. For instance, one platypus X carries the DMRT1 gene, which is found on the Z chromosome, the male determining one, in birds. It's like a living evolutionary mosaic. And we can't forget that even when sex chromosomes exist, the autosomes, the non-sex chromosomes, can still play a critical role. Take Drosophila, the fruit fly.
Right, they have X and Y, but it works differently.
It does. In Drosophila, sex isn't strictly determined by the presence or absence of the Y chromosome like in mammals. Instead, it's determined by the ratio of X chromosomes to the number of sets of autosomes. It's called the XA ratio. A ratio of 1, like 2 X's and 2 sets of autosomes, XXAA, makes a female. A ratio of 0.5, like 1 X and 2 sets of autosomes, XAA, makes a male. The Y chromosome in flies is needed for male fertility, but not for determining maleness itself.
So it's a balance, a genomic ratio, not just one master switch gene.
Exactly. It reminds us that sex determination is ultimately a complex developmental process, often controlled by numerous genes interacting across the entire genome, not just a single locus on a sex chromosome. And the fact that sex determination can shift from strict chromosomal control to being entirely dictated by external cues, well, that just highlights the incredible power of gene-by-environment interactions. In many organisms, the environment literally acts as the master switch for sex.
Right, moving beyond the genome itself. And some of the behavioral or environmental cues that trigger sex determination are truly remarkable. The sources give some great examples, like the Osedax tube worms, those bizarre deep-sea creatures that live on whale bones.
Oh, the bone-eating worms. A fantastic example. If an Osedax larva happens to settle directly onto the whale bone in the deep sea, it develops into a relatively large female, which houses symbiotic bacteria to digest the bone.
Okay, female on the bone.
But, if a larva happens to land on an already established female, instead of the bone, it develops into a tiny, microscopic, parasitic dwarf male that lives inside the female's tube. Same genes, totally different outcome based purely on settlement location.
Wow, environment as destiny almost.
Yeah.
And there's a similar story with the green spoonworm, Bonellia viridis, right?
Very similar logic. If a free-swimming Bonellia larva lands on the open sea floor, it develops into a large female, maybe 10 to 20 centimeters long, with a long proboscis. But if that same larva happens to land on the proboscis of an existing female, chemical cues from her cause it to develop into a tiny 1 millimeter long parasitic male that lives inside her reproductive tract. Again, the environment dictates the sex.
Incredible. And we can't possibly skip the rather dramatic penis fencing flatworms described in figure 13.7.
Ah, yes, the pseudo-serrated flatworms. These are simultaneous hermaphrodites. They have both male and female reproductive organs. But their functional sex role during a specific mating encounter is determined by behavior.
How does that work?
They engage in this fencing duel, using their penis-like stylets. Whoever successfully manages to pierce the skin of the opponent first injects sperm, effectively acting as the male for that encounter. The loser, the one who gets pierced, takes on the female role, investing resources in developing the eggs. Behavior determines functional sex in the moment.
Fascinating. So beyond these location and behavioral cues, temperature is a huge factor too, isn't it? Especially in reptiles.
Massive factor. In many reptiles, like alligators and many turtles, the temperature of the nest during a critical period of embryonic development determines the sex of the offspring. There aren't sex chromosomes doing the job. It's purely the incubation temperature. Hotter might mean males, cooler females, or vice versa, depending on the species.
And social cues can be just as powerful, particularly in fish.
Absolutely. Many tropical reef fish exhibit sequential hermaphroditism. They change sex during their lifetime, based on social hierarchy. In some species, the loss of the dominant male in a group will trigger the largest dominant female to physiologically change sex and become the new male, often very rapidly.
So the social environment triggers a complete physiological transformation.
It does. In other fish, like clownfish, it can even work in reverse, or relative size within the group dictates who changes and in which direction. It's all about social context.
And this links back to resource allocation too, doesn't it? Especially in species with that haplodiploidy system, like bees, wasps, and ants. Can you remind us how that works?
Sure. Haplodiploidy is common in the Hymenoptera. In these insects, unfertilized eggs develop into haploid males, they only have one set of chromosomes, while fertilized eggs develop into diploid females, with two sets of chromosomes.
So the mother controls the sex by choosing whether or not to fertilize the egg.
Precisely. And she can use environmental cues to make that choice strategically. The source mentions the parasitoid wasp, Lariophagus distinguendus. This wasp lays its eggs in grain weevil larvae. What the female wasp does is assess the size of the host larva. If it's a large, well-nourished host, she'll lay a fertilized egg, a daughter, because the female offspring needs more resources to grow large and produce lots of eggs herself later.
Makes sense.
But if she encounters a small, poor-quality host, she'll lay an unfertilized egg, a son. Males need fewer resources just to develop and produce viable sperm, so she's actively choosing the offspring's sex based on the predicted resource availability. It's resource-dependent sex allocation.
Right. And this powerful environmental influence, it's not just limited to determining sex, of course. It shapes countless other traits.
Oh, absolutely not. The environment dictates innumerable phenotypes, constantly interacting with the genotype. It really drives home the point that a genome isn't some isolated blueprint operating in a vacuum. Think about hydrangea flowers. Their color is directly influenced by the pH of the soil they grow in. Acidic soil, blue flowers. Alkaline soil, pink flowers. Same genes, different environment, different look.
Or even fur color in some mammals.
Yeah, like Siamese cats or Himalayan rabbits. The fur on their extremities, ears, paws, tail is darker because those parts are slightly cooler. And the enzyme responsible for pigment production is temperature sensitive. It only works effectively at lower temperatures. And critically, think about human height. There's obviously a strong genetic component, but the eventual stature someone achieves is heavily influenced by diet and nutrition, especially during development.
The sources mention the post-World War II Japanese generation as a key example of this.
Exactly. That generation saw a marked population-wide increase in average height compared to their parents, primarily attributed to major improvements in nutrition and public health after the war. Same gene pool, different environmental inputs, dramatically different average phenotype.
And this flexibility, this ability of a single genotype to produce different forms or life stages in response to the environment, that has a name, right? Phenotypic plasticity.
Yeah. Phenotypic plasticity, precisely. It's the ability of one genotype to produce more than one phenotype. Maybe a different morphology, physiology, or behavior, sometimes called a morph or ecomorph, in response to specific, often facultative, meaning optional or conditional, environmental cues.
And it's seen as evolutionarily significant.
Hugely significant. Phenotypic plasticity is often viewed as a potential crucial first step in adaptation and even speciation, particularly sympatric speciation, where new species arise without geographic isolation. A plastic response might allow a population to utilize a new resource or habitat, and if that becomes advantageous, selection might later favor genetic changes that stabilize that new phenotype, making it less dependent on the environmental cue. And while we're wrapping up sex-linked complexity, we should probably briefly touch on Haldane's rule, which the source mentions in an endbox.
Okay, what's Haldane's rule?
It's an observation about hybrids. When you cross two different animal races or closely related species, if one sex among the first generation, F1, hybrid offspring is absent, rare, or sterile, that sex is almost always the heterogametic sex.
Meaning the one with two different sex chromosomes?
Exactly. So in mammals, that would be the XY males. In birds and butterflies, it would be the ZW females. This pattern is often thought to be linked to the expression of deleterious recessive alleles on the sex chromosomes that get unmasked in the heterogametic sex.
Interesting pattern. Okay, so despite all these complex determination systems, environmental influences, plasticity, many species still manage to maintain that remarkably stable 1.1 ratio of males to females. How does that balance hold?
That stability generally comes down to a concept called frequency-dependent selection, often referred to as Fisherian dynamics, after R.A. Fisher, who first mathematically described it. The logic is quite elegant. Imagine a population where, for some reason, males become rare. Any individual whose genes predispose them to produce more sons will suddenly have a huge reproductive advantage because their sons will have relatively little competition for mates.
Their genes for producing sons spread rapidly.
Exactly. Those genes quickly increase in frequency, which brings the sex ratio back towards 1.1. Conversely, if females become rare, genotypes producing more daughters gain the advantage. It's a self-correcting mechanism. Whichever sex is rarer automatically becomes more valuable reproductively, driving the ratio back to the equilibrium point, which is usually 1.1, #DEG_outro. So to kind of synthesize this whole deep dive, what the sources really drive home is that inheritance is this incredibly complex, dynamic matrix. Mendel's elegant rules are truly just the foundation. They get profoundly modified by gene-gene interactions like epistasis, by non-nuclear factors passed down, usually maternally, through the egg cytoplasm, and maybe most dramatically by the environment itself, manifesting as phenotypic plasticity, and even determining fundamental traits like sex.
Right, so the core message for you, the listener, is really this: that immense underlying potential for genetic variability. Remember those 10 billion combinations from just 100 interacting genes, combined with the ability of organisms of single genotypes to flexibly respond to their surroundings. That is the fundamental engine that fuels all adaptation and the success of evolution over time.
And maybe here's a provocative thought stemming from those Y chromosomeless rats to leave you with. The fact that organisms like the Japanese spinous rat have completely discarded their Y chromosome, rearranging sex determination onto other parts of the genome, suggests that even structures we think of as fundamental pillars of genetic identity are surprisingly fragile and constantly evolving. It really makes you wonder, what other crucial elements of our own genome, things we currently consider immutable bedrock, might actually be subject to eventual loss or radical evolutionary change over the vast expanse of deep time?
That is a truly mind-bending thought to end on. Evolution never stops tinkering. Thank you so much for joining us on this deep dive into the fascinating complexities of genes, environment, and inheritance. We'll catch you next time.